Today we are going to deal on a most important topic, so that is growth and nutrition of, so that is bacteria and the microorganisms. Here, the growth and nutrition of microorganisms includes the bacterial growth and bacterial nutrition. In that, the first we are going to deal about the bacterial growth. Here, the bacterial growth is nothing but it is the increase in size of, so that is bacteria or microorganisms. The bacterial growth it includes, so that is the following steps. So that is bacterial cell division, the generation time, the bacterial count, and the bacterial growth curve. Okay, so let us see one by one detail. Here now about the bacterial cell division. Here the reproduction takes place in the bacteria in the form of binary fission. So that is one cell is divided into two, two cell divided into four, four into so 8, and okay, so likewise, so the binary fission, it means one cell a division, that is what we call as the bacterial cell division. Here the bacterial cell division usually takes place by the process we call as bacterial or binary fission. Right, now coming to the generation time. The generation time is nothing but it is a time taken by a microorganisms or the bacteria to produce or to multiply. So that is what we call it the generation time. Generation time is nothing but it is a time taken by microbes or bacteria to divide one cell into two cells. Okay, it means start dividing. So that time would be called as generation time. Here all medically important bacteria having a, so there is a, okay, so there is generation time about 20 minutes, almost all bacteria. But here the Mycobacterium tuberculosis has it is having a, so that is 20 hours, 20 hours. It is having a, it takes 20 hours to multiply one bacteria into, so that is two bacteria. The Mycobacterium leafrem, it is having a the 20 days, that is generation time. Here the different microorganisms having the different generation time. Now come the bacterial count. Here the bacterial count is nothing but the number of, so the microorganisms or number of bacteria in a particular specimen. For example, if you are going to a culture as, so that is my bacteria in a culture container, you are pouring as, so it is blood. This contains the culture media. This media is very important to grow the bacteria. Here the bacterial count, it means it includes, so there is whole numbers, so whether death bacteria as well as live bacteria. Here in that, so the total number, total cell count as well as viable cell count. The total cell count is nothing but the number of, so that is bacteria living in a, so that is particular, so the specific culture media. In that viable is nothing but the number of living bacteria in a particular specimen, particular culture media. So that is what we call as cell count. Now come to the bacterial growth curve. Here the bacterial growth curve, in a particular, so the culture media, the bacteria grows. Okay, so take time and grows rapidly and maintains constant, and again it declines. Okay, so it is display in there, so that is one graph, so that is in curve. So that is what we call as a bacterial, so the growth curve. The bacterial growth curve, so here the number of, so that is bacteria is growing, it taking time, how much time it's taking. Here it is divided into four phases or four stages. The first stage, so that is lag phase, second phase log phase, third phase stationary phase, and fourth stage is the decline stage. Now come to the, so there is lag phase. Okay, so lag phase. The lag phase is nothing but it is also called as the initial phase. It is the time taken by a bacteria to adjust to the new environment and to grow, okay, so effectively. So that is what we call as a lag phase. The lag phase also falls in this initial stage, or so that is first stage. Here the bacteria doesn't doesn't go under the multiplication. It takes the one's time to adjust to the new environment. And yesterday only in the, so the 2020 World Cup match happens. Here the first two over, so that how much they did runs, so that is only 12 they did, because they taken time to adjust to this, so that is bowlers. Okay, so again that is what we call as, so the lag phase. In the log phase, the bacteria already adjusted to that environment, it started to multiply. The sudden rapid, so that is the multiplication of bacteria take place, there is suddenly the increase in the number of, so that is bacteria take place. That is we call as an, so that is log phase or exponential stage. It means sudden, so there is increase in the number of, so that is bacteria takes place. Okay, so ಆಮೇಲೆ so after 4 hour after 2 hours what they did, okay, so 150 runs. Okay, so the run becomes very increasing because already they are adjusted in the log phase only. So in the log phase, in the exponential phase, suddenly so they increase their numbers. In the same way, the bacteria also, so that is does like that only. So already in the log phase, they are adjusted to the new environment. It start multiplying. It increases numbers rapidly. So that stage we call as a log phase or a exponential phase. Now, come to the stationary phase. In the stationary phase, what happens in the so the culture media? So already, so the bacteria are multiplying, and also in the same process, the bacteria also start dying. Here the number of bacteria, the multiplying is equal to number of the bacteria are going to die. The just number is remains same, so that is we call as a stationary phase or a constant phase. Here it means due to this, so there is shortage of the nutrient in aquaculture medium. The number of growing bacteria is equal to number of dying bacteria. So it constant number. So that is what we call as a stationary stage or a so the stationary phase or a so the constant phase. Now coming to the last one is so there is decline phase. The decline phase also we call as a death phase. It means due to this, so that is insufficient nutrient in a culture media, the bacteria is going to die. Here the number of living organisms, so that is bacteria, so that is decreases. Number of death organisms increases. It means that is total count. The total count increases, the viable count decreases in the decline phase array, so that is death phase. Okay, so this is regarding to what? So that is bacterial growth. Under this bacterial growth, the bacterial cell division, generation time, the bacterial count, that is total count and viable count, and the bacterial the growth curve. Here bacterial growth curve is divided into, so there is 4 types: the log phase, stationary phase, and the decline phase. Here in the bacterial growth is nothing but the growth occurs to bacteria. Okay, so this is regarding to what? So that the bacterial growth.